Welcome for this session. Okay, so we will be performing experiments regarding the membrane transport. Okay, so we will try to demonstrate transport of molecules across the cell membrane. As discussed in your lecture, okay, so homeostasis pertains to the maintenance of a constant internal environment in a cell or organism. So this is very important in maintaining a stable environment for all our enzymes and other molecules, okay, to function at their optimum level. Okay. Now, the movement across the plasma membrane can either be a passive transport or what we call diffusion, wherein there is no need for energy, particularly ATP or adenosine triphosphate. So this can either be a simple diffusion or a facilitated diffusion. So as mentioned in your lecture, simple diffusion, you don't really need other molecules to help in the transport of substances across the membrane. So the substance just move from an area of higher concentration to an area of lower concentration. Whereas in facilitated diffusion, there is still no need for uh, ATP or other source of energy, but we do use some carrier or protein channels in order to help in moving the molecules from one area towards the opposite side of the cell. But again, it has to follow, okay, the principle of moving from a higher concentration to a lower concentration. Compare that to an active transport, okay, wherein you are moving molecules against the concentration gradient. So from lower concentration to higher concentration. Since you are moving against the concentration gradient, you will need a pump. And this pump would usually require an energy source. It can either be in the form of ATP or other phosphate containing energy currency of the cell, or it can be riding on another molecule, okay, like sodium, okay, so that it would be able to transfer, okay, to the other side of the compartment. And then for bulk transport, okay, this will now pertain to massive amount of substances being moved from from one side of the cell to the other, either from outside going into the cell or from inside the cell going out of the cell. So if there are molecules involved, then this is, if there are large molecules involved, then we call this phagocytosis. But if this is predominantly uh, water, okay, with some suspended molecules, then we call this pinocytosis. Now, going back to diffusion, okay, we want to again define it as the spontaneous movement of molecules, okay, of different substances, okay, and this results from the kinetic energy of the molecule. So this is what we call the Brownian motion, wherein one molecule will have this innate energy wherein it would vibrate so that it will bump, okay, onto the surrounding molecules. So the closer they are to each other, the more that they bump into each other. And movement of molecules is from, as we have mentioned, from a region of higher concentration to a region of lower concentration. And this process of movement will continue until the substance is equally distributed throughout the medium. So once the molecules are now equally distributed throughout the medium, it doesn't mean that the movement will stop. It will just mean that if one molecule, for example, moves to the right, okay, it will be replaced by another molecule moving to the left. So there will be continuous movement, but the spread of the molecule in the medium would more or less be uniform. So as exemplified by this uh, video. So as we can see here, starting from an area where the molecules are clustered together, okay, slowly they bump into each other and then they start spreading out. Okay, so this is because of the kinetic energy, okay, present in each of those molecules, and as they bounce against each other, they now push each other away, so that they would now have equal spaces in between them. So next we have another type of membrane transport, okay, called osmosis, which is a special form of diffusion. So it is the movement of water from a dilute solution, meaning of lesser concentration, okay, to a more concentrated or hyperosmotic, okay, solution through a semi-permeable membrane or a selectively permeable membrane. So this type of membrane, okay, allows only certain molecules to pass through, and the process of diffusion of crystalloid solutes through such membrane, on the other hand, is known as dialysis. So you may be familiar with the procedure, the treatment procedure done on patients with problem in eliminating toxins in their body, especially those with kidney failure. So they undergo dialysis. Okay, so the process that is used in dialysis is osmosis. So I will show you a clip, okay, demonstrating osmosis. As we can see here, we have a semi-permeable membrane, and then on one side of the beaker or compartment, you have purely water molecule. And on the other side of the semi-permeable membrane, you have a mixture of water molecule and carbohydrate sugar molecule. Okay. Now, since the sugar molecule, okay, or the glucose cannot pass through the semi-permeable membrane, okay, and the two compartments, okay, are somewhat connected to each other, it would want to equalize, okay, its concentration. So in order to decrease the discrepancy or the difference between the concentration of glucose on this side, okay, on the right side compared to the left side of the beaker, okay, so water will now be the one to pass through the semi-permeable membrane because it is allowed by the membrane. Okay, so by doing that, we are diluting, okay, the glucose solution, okay, hoping that the concentration of the glucose solution on both sides will equalize. But then, of course, because in this sample, there is no glucose on one side, that would be an impossibility. Okay, there's no way for this particular setup to really reach a true equilibrium. Because after some volume, okay, another force, your hydrostatic force, which is the force of water being pushed out, okay, as pulled by gravity, will now take over the urge of this compartment to attract water towards it. Okay, so in your lecture, you will discuss more about this, especially in the circulatory system. Now, moving on to the objectives of our laboratory activity. So, at the end of this activity, the student should be able to describe the processes involved in diffusion and osmosis, relate factors such as molecular weight, temperature, and molecular size with the rate of diffusion, and describe the role of the plasma membrane in the transport process. For the materials that will be used in this laboratory, okay, we will just discuss them as we go on with each part of our activity. Okay, so let's first look at our exercises, okay, demonstrating diffusion. So first, okay, we will try to look at the relationship between diffusion and molecular weight. So in this activity, the student will investigate the relationship of molecular weight and diffusion rate. So in part 1, we have this setup. Okay, so you can, as you can see, we have a clean glass tube, okay, clamped to horizontally, okay, to an iron stand, okay, and then at the end of the tube, you have their cotton buds, okay. So in these cotton buds, okay, so we will be adding simultaneously, okay, approximately 10 drops of 1 mole of HCl on this side, and then 10 drops of ammonium hydroxide on this side. So take note that the molecular weight of chloride, which is part of your hydrogen chloride, is 35 moles, millimoles, whereas for your hydrogen ammonia, which is part of your ammonium hydroxide, is 18. Okay? Now, so after we have added simultaneously, okay, those two reagents on each, okay, end, then we will now start observation. So if we were to do this, okay, in the laboratory, one of the main very important precautionary measure that we observe in the laboratory is that we are careful when it comes to hydrochloric acid and ammonium hydroxide, because these are both uh, very caustic or uh, volatile uh, reagents. Okay, so they can cause severe burns, so and damage to your clothes. So in case you in the future you directly handle these two reagents, okay, remember to be careful, okay, when you handle these two reagents, and always ask your professor, okay, where to place, okay, the discarded material, so that you would not accidentally discard it in the general waste bin. Okay, that's very important safety precaution in the laboratory. So after we have placed, okay, hydrogen chloride on this end, and then ammonium hydroxide on the other end, okay, we will observe the tube, okay, for a white ring precipitation that will form inside this clear glass tube, okay. And then using a ruler, we will now measure the distance from each end of the tube up to the white ring, and then we will record the data in centimeter, okay. For us to have a better idea of how this is done, okay, I will show you a video demonstration. So we have our setup. We are now going to place, okay, equal amount, okay, of drops of the two reagents on each cotton bud, okay, simultaneously. So as I have mentioned, please be very careful when handling this reagent, okay. Try to avoid returning the excess reagent into the reagent bottle to avoid contamination. Oops. Now we're going to plug it in simultaneously so that the molecules of each reagent will start to diffuse simultaneously towards each other. So hydrochloric acid will be diffusing from this end towards the middle, and then the ammonium hydroxide will be diffusing from this end towards the middle. Okay? And then wherever they will end up meeting each other, okay, they will now form a white precipitate. Okay? So that white precipitate is a combination, okay, is a chemical reaction between your hydrochloric acid and ammonium hydroxide. So as part of your activity sheet, okay, you need to show, okay, your balance equation of the reaction between your hydrochloric acid and ammonium hydroxide. Now, after a few minutes of waiting, okay, we are now able to observe the formation of the white ring precipitate. So, can you see it on the screen? Can you see the white precipitate? Okay, so if you are not able to see it, here it is. Okay, so that is where the white precipitate ring formed, okay, after we have allowed your hydrochloric acid and your ammonium hydroxide to diffuse through this clear glass tube. And as for the distance traveled by both reagent, okay, take note that the length of this entire glass tube is more or less 540 centimeter. Okay? Because by knowing the full length of this glass tube, okay, it would be easier to measure the distance traveled by this side by just measuring the shorter distance. Okay? So again, the full length of your glass tube is 540 centimeter. Okay. So now we are on the left side. Okay? The hydrochloric acid side. So you can see that this is the end of the glass tube, and then here is the white precipitate rate. So what is your measurement? Okay, can you see that? So knowing that this is the distance traveled, okay, by this reagent, okay, what is the distance traveled by the other reagent? And what, okay, is the chemical, okay, product of this white precipitate ring? Based on our results, what can you say about the molecular weight of the two reagent, okay, in relation to the distance traveled? So since we have distance over time, okay, so that would be the rate of diffusion. So what is the relationship of the rate of diffusion to the molecular weight of our reagent? Now let's have another experiment demonstrating the relationship between rate of diffusion and molecular weight. So here we have a Petri dish, okay, and we filled it up with agar, okay. So in the agar, okay, we place some holes, okay, so we bored, okay, 3 equidistant, okay, holes in our agar plate, okay, and then in each of these hole, okay, we will now be placing 3 different reagent. So in one hole, okay, so we will be placing 1% Congo red, and the other, okay, hole, we will be placing 1% methylene blue, and in the other, or rather in the other hole, we will be placing 1% potassium permanganate. So take note of the molecular weight of these reagents, okay, because we will need that later in explaining the relationship of the molecular weight with the rate of diffusion. Okay, so I will now show you the video, okay, demonstrating how we perform this experiment. So this is already a speed up demonstration. So basically, we place Congo red on one hole, potassium permanganate on another, and then methylene blue on another. And this is a time-lapse video. So you can see each reagent slowly diffusing, okay, to its surrounding. So it will take about 30 minutes to finish this exercise. Okay, then after 30 minutes, we will measure the diameter of the colored ring, okay, around the holes in millimeter. And then we will record them on our activity sheet. Okay, so this is a close-up of our agar plate after 30 minutes. So do you remember this hole contains your 1% Congo red, and we have placed the 0 mark, okay, of the ruler here, and then of course we are using the metric system. So more or less, okay, this is the edge. Okay, this is the other end of our diameter. This is the other edge of our diameter. So how big is the diameter, okay, of the diffusion of your Congo red? And then here in the other hole, we have our 1% methylene blue. So we place 0 on this side. And then on the other side, this is the edge of our diffusion. Okay, so what is the number? What is the measurement that we got from this experiment? And then of course, for potassium permanganate, so this is the edge, okay, one edge of the potassium permanganate, and then this is the other edge of the diffusion of potassium permanganate. So what is the distance? Okay, what is the distance that we got, okay, from this experiment? So by comparing the measurements that we got from the three reagents and correlating it with the molecular weight of each, okay, so what do you think is the relationship between the rate of diffusion and molecular weight among these three reagents? Now let's move on with the demonstration, okay, of the relationship between rate of diffusion and temperature. So for this etap, okay, so we have our hot plate for heating up water. And then we also have here ice cubes, okay, so that we will have cold water. And then we have two thermometer which will now register the temperature of our water in each respective beaker. And then here in this test tube, we have our granule reagent. So for this experiment, we will be using potassium permanganate granules. So now let's look at the video, okay, demonstrating how we perform this experiment. Okay, so in one beaker, okay, we filled it, okay, halfway, half full, with cold water, Uh, and you can see that it is 17 degrees centigrade. And then in the other beaker, okay, we filled it up with hot water, okay, and the temperature of the water is 50 degrees centigrade. So one is cold, the other one is hot, okay. Now we place both beakers in an area where they will not be disturbed, okay? And then we slowly added the same amount of potassium permanganate crystal, okay, in each beaker. So make sure that the amount of potassium permanganate are more or less the same for each beaker. So this is now a time-lapse video. So can you guess which beaker contains the cold water and which beaker contains the hot water? So after 30 minutes, okay, we observe the changes in the distribution of potassium permanganate, and then we record our observation. Okay. So this is now our result. Okay, so the beaker on the left is the cold water, whereas the beaker on the right contains the hot water. So what can you say about the relationship of the diffusion of the potassium permanganate crystals, okay, in relation to the temperature of the water? Next, we will look at the relationship between rate of diffusion and the molecular size of our reagent. So in this part of the activity, okay, we will be using a selectively permeable membrane using our cellulose membrane. So this is an artificial cellulose membrane, okay, and this will now be used to represent our cell membrane. The surface of the membrane contains tiny pores that will allow certain molecules to pass through it, but not the others. So it's going to be selective. The experiment, okay, will use starch and iodine solution. So as we may have known already in our general biology in high school, the reaction between the two substances, okay, between your starch and iodine will result in a blue-black discoloration, which will indicate the presence of both in the medium. So for the procedure, we have here our 50 ml beaker, and we fill it up with water up to a third, okay, of our beaker. And then we place, okay, we add 4 drops of our potassium iodide, okay, so that will now serve as our indicator, okay, in case there is now the presence of starch. Now we set this aside, okay, and then we now proceed with our cellulose sac. So we have here our cellulose sac that has already been placed in water to loosen it up. One end of the cellulose sac has already been tied with a string, and then we fill it up with 1% starch solution. After we have placed the 1% starch solution, okay, in our cellulose sac, we secure the other end of the cellulose sac, okay, with another strand of tie, okay, so that our starch solution will not escape. Now, make sure to rinse the outside of the cellulose sac so that no starch solution will be present, okay, outside the cellulose sac. Okay, so there will be no traces of starch outside the cellulose sac. So after that, okay, we now place the cellulose sac in our beaker that we have already prepared, okay, and then we let it stand for 20 minutes. And then we will check for color changes, okay. So let's now observe what happened to our experiment. So we have our beaker with the water and potassium iodide. And here we have our starch solution inside the cellulose. So what happened? So where did the blue-black discoloration, occur? So can you tell me where the blue-black discoloration was observed? Was it observed inside the cellulose sac or outside the cellulose sac within the beaker? Okay. Knowing our result, so can you now tell me which substance, was able to pass through the cellulose sac? Was it starch or was it potassium iodide? Okay, if you would compare now the size of starch molecule to potassium iodide molecule, which one is larger and which one is smaller? So with that, can you tell me the relationship of the molecular size to the rate of diffusion, or in this case, the ability to undergo diffusion? We are now done demonstrating our activities regarding rate of diffusion. Let's now look at the factors that can affect the rate of osmosis. For our first activity, the rate of osmosis will be investigated using artificial semi-permeable membranes. So our ever-dependable cellulose sac. Okay? So the cellulose membrane, is also known as dialysis tubing, as we have explained before. Now, we will need 5 pieces of this cellulose sac, but before that, we also need to prepare, beakers, So in these beakers, for beakers 1 to 4, which is labeled here as A, B, C, and D, we place distilled water, And then for the last one, the last beaker, we place here 1 mole sucrose solution. So we fill it up, up to about 2/3 full. So for 1 to 4 or ABCD beaker, you have distilled water. For the last beaker, it would now contain 1 mole of sucrose solution. Now, let's now proceed with filling up our cellulose sac. So we will now fill up the cellulose sac with our designated solution. So for sac 1, we will place distilled water, 10 ml of distilled water. Sac 2, we will place 0.4 mole of sucrose solution. Sac 3, we will place 10 ml of 0.8 mole sucrose solution. And then for sac 4, we will place 1 mole sucrose solution. And then for the 5th sac, we will place 10 ml of distilled water. So when we fill it up with the designated solution, we have to make sure that all the bubbles are removed, because we don't want to have bubbles because it can interfere with our result later on. And after that, we will securely tie, the top of each sac with a string. So more or less, this is how it will look like. So after we have filled up, our cellulose sac with our 10 ml designated solution, we will weigh, our cellulose sac. So the weight of our cellulose sac, that will now be our initial weight. So that is the initial weight. After which, we will immerse it in our prepared beaker. So remember, beakers 1, 2, 3, and 4 contains distilled water, while beaker number 5 contains 1 mole sucrose solution. And then for the cellulose sac, okay, so cellulose sac 1 has distilled water. Then we have 0.4 in the cellulose sac 2, 0.8 in cellulose sac 3, 1 mole in cellulose sac 4, and then distilled water in cellulose sac number 5. Okay. We will then allow this to stand for 30 minutes, after which, okay, we will remove the sac and then wipe off the excess water, okay, outside the sac, and then wait again. Okay, so this weight is now going to be the final weight of our cellulose sac. Okay, so after weighing all the sacks, so here is our result. So for setup A, okay, so the sac, cellulose sac, okay, 1 or A, had an initial weight of 4 grams and a final weight of 4 grams. For cellulose sac B or number 2, it has an initial weight of 5.2 grams and a final weight of 5.3 grams. And then for cellulose sac C or 3, we have an initial weight of 4.6 gram and a final weight of 5.4 gram. For cellulose sac D or number 4, we have an initial weight of 4.6 gram and a final weight of 5.2 gram. And then for our cellulose sac E or number 5, it had an initial weight of 4.4 grams and a final weight of 3.5 grams. So knowing our initial and final weight, we will now compute for the percentage change in weight using this formula. So change in weight would be equal to final weight minus the initial weight divided by the initial weight, multiply that by 100%. So whatever is the percentage change in weight that you get, okay, you will then graph, okay, this percentage change, okay, and then so that we would be able to see the trend, whether our setup, okay, our cellulose sac gained solution or did it lose solution. Now, for our last activity, this will now involve, okay, red blood cell and osmosis. So, in this part of the activity, okay, we will allow students to observe osmosis in red blood cells. So, if this was face-to-face, okay, we will have a lot of you students as our guinea pigs. But for this exercise, okay, so we just had a demonstration. So, basically, when performing this setup, we will need 3 microscopes, okay, and then we will prepare 3 clean dry slides and 3 cover slips. Then label each, okay, slide, one with 0.9% sodium chloride. The other slide, we label it as distilled water. And the third slide, we label it as 5% sodium chloride. Okay? So make sure to label them because all of this would look the same. Okay? In our naked eye. Now, so when you are going to prick someone, okay, make sure that your hands are clean. Wash them, disinfect. And then we will now prepare a sterile lancet. So here we have a lancet pen, and then here we have our disposable lancet. So we place this inside the pen, okay? And then we can calibrate this so that we will have a certain length, okay, coming out of our pen, okay, so that our subject will not have any unnecessary, okay, harm. So we can choose either the middle finger or the ring finger of our subject, preferably the non-dominant hand, okay, so that in case you still need them to sign some important papers later on, they can still do so, okay. Now, so we prepare the fingertip by wiping, okay, with alcohol, okay, cotton with alcohol or alcohol swab, and then you allow it to dry, okay, before we prick the finger. Now, position the lancet, okay, opening against the fingertip and press the release lever until it clicks. And then once it clicks, okay, after some gentle squeezing of the finger, okay, from the base of the finger, a drop of blood should come out, okay, from our needle prick. Now, apply the drop on the labeled slide. So, one drop on each of the slide. And then, on each slide, okay, we place a drop of the respective reagent. So, if the label says 0.9% sodium chloride, you add, okay, 0.9% sodium chloride uh, on that slide. Okay, and if it says distilled water, we add a drop of distilled water, and then we place our cover slip on top of it. Okay, and then we examine it under the microscope. Okay, so we need to do this a bit fast, okay, so that we will not end up with a dried up specimen. So we don't want to have a dried up specimen, and drying can also change the concentration of the solution that we have applied on our red blood cells. So let's now look at the appearance of the cells that we have added, okay, different concentration of solution. So here we have our red blood cell, okay, in 0.9% sodium chloride solution. So what can you say about the morphology of our red blood cell? So what is the shape? So this is another view, okay. This is now still your 0.9% sodium chloride solution. This time we have red blood cell in distilled water. So what can you now say about the shape of the red blood cells? And what can you say about the quantity of the red blood cells? So we can even see some white blood cells, okay, floating around together with our red blood cells. So what do you call, okay, what do you call this what is the description for these kinds of red blood cell? Okay, this time around, well, we don't have the 5% sodium chloride solution, but 2.5 sodium chloride solution is still higher, okay, more concentrated compared to your 0.9% sodium chloride solution. So, what can you say about the morphology of your red blood cell when they are now placed in your uh, 2.5 or 5% sodium chloride solution? So among the three, your distilled water, 0.9% sodium chloride solution and your 5% sodium chloride solution, which among them are said to be your hypertonic solution, hypotonic solution, and isotonic solution? And how do they affect the red blood cell? What happened to the red blood cell when placed in these types of solutions? So that's it for our laboratory activity on membrane transport. So with the data that we have generated, okay, so interpret, okay, our data, and then draw your conclusion, okay. Explain or give the relationship between the different factors and the rate of diffusion or the rate of osmosis. And then you have your laboratory answer sheet. Okay, so please answer the guide questions, okay, provided in your laboratory answer sheets. Thank you.